All right, folks, hope you're doing all right. We're going to do a little video to summarize this process that you all know and love. And at GCSE, you were told that these two bad boys get together and they form this legend and this beautiful gas. And you know, you can balance it with your sixes. 6, 6, 6. Now, that's a little bit of a fib. It's a lie, because that's one single reaction. And photosynthesis is not one single reaction. It's many, many reactions. And we call many reactions with reactants that form products. And the products then go on and form more reactants to form more products. So we call this a metabolic pathway. And photosynthesis and respiration are classic examples of beautiful metabolic pathways where there are reactants and there are products. And those products go on to start the reactions of new pathways. And it goes on and on and on. And sometimes the products that are made in a pathway inhibit a reaction as well. So let's have a look at photosynthesis, and there are two parts to it. There is the light dependent and there's the light independent. Okay. And the key to photosynthesis is that it happens in an organelle, which looks a bit like a rugby ball. There's the outer membrane. And here's the inner membrane. Look how I draw it. It's kind of folded into turrets, layers upon layers of flattened membrane. If you could imagine it was 3D, it'd be coming out to get you, because it's in 3 dimensions like that. Okay. And that inner membrane is called the thylakoid. And the stacks of the thylakoid are called the grana. Okay. And there's a tiny little bit of DNA in there. Bit of DNA. And there's acyl ribosomes as well. So there's a real clue there that maybe in Yieldy past, maybe the chloroplast was a separate organism. Looks like a little face, doesn't it? Looks like ET. Okay, let's start with the light-dependent reaction. So we're going to look really close up at this membrane in here, okay? So as you know, when you look at a membrane, it's got a good old phospholipid bilayer, and that's given a little bit of stability with cholesterol. And then you've got these chunky proteins that stretch all the way across, and they're called intrinsic proteins. Now, this is what's absolutely amazing about the grana, is underneath these intrinsic proteins on the thylakoid, you've got these primary pigments, and these primary pigments are pigments like chlorophyll. So you get chlorophyll A and you get chlorophyll B, but there's many more. And then surrounding the primary pigments, I'll draw them as little triangles, you've got these accessory pigments. You want to call them secondary, don't you? But they're called accessory pigments. And they've got chemical identification like xanthophyll and carotenoids. Carotenoids. Okay, carotenoids. And they absorb different wavelengths of light. In fact, all of these pigments absorb different wavelengths of light. And different plants will have different combinations of these or different proportions of these to maximize the wavelength of light that is absorbed. Now, there is a difference. There's a big difference between primary and accessory, because primaries get the electrons excited when light hits them, and then those electrons are released to a higher energy level. Okay. And because the electrons are leaving the pigment, the primary pigment is photoionized. Okay. So how would you photoionize a primary pigment? shine light of the right wavelength on it, and those electrons will get excited and then they'll be released. Please use both those terms, all right? Now, if the light hits the accessory pigments, it absorbs the energy, okay? But all it does is it passes the energy on to the primary pigments. And it's the primary pigments that get excited and released. The electrons are released to a higher energy level. So let's put the side of the paper as high energy, all the way down to low energy, like that. Now, there's two types of primary pigments. We're going to call them PS2 and PS1. And they're found in the membrane. Okay, so these pigments are found underneath those intrinsic proteins in the membrane. So when light comes along and hits those chlorophylls, the electrons get excited. That's what I'm doing to get the electrons excited. Can you see I'm drawing them round and round? That's excited electrons. And then they rise to a higher energy level. So there they go. I'm going to put in 4 electrons. I'll tell you why in a minute. And they're accepted by a protein, an electron acceptor protein. And that electron acceptor protein is found in this membrane. Okay. Very good. Could be there. Electron acceptor. So the electrons, whoosh, they're passed on to the electron acceptor. When the electron acceptor gains the electrons, it becomes reduced, and the chlorophylls in PS2 become oxidized or photoionized. So we've got to deal with that. We can't have our chlorophylls being all positively charged. So what happens at the same time, I'm going to give 2 molecules of water here, but it doesn't matter. There's 2 molecules of water, and the light is hitting the water, and water gets undergoes photolysis. So what's that? Well, it basically ionizes into 4 hydrogen ions, 4 electrons, and a molecule of O2. That O2 is released, and it's breathed in by all you lot, and you're happy. It's kind of happy. But we need to replace these 4 electrons that are excited and leave. So there they are. They're replaced by those. Happy days. Now these electrons that have been excited and released then get passed down a series of electron acceptor proteins. And as they get passed down those electron acceptor proteins, you can see from the diagram that they're moving down our energy levels. So the energy is being transferred. Now, on a very simplistic level, you can say that energy is being transferred to a molecule of ADP plus P, which makes ATP. Okay, on a very simplistic level, that's where you can say that energy is transferred to. And I'll come back at the end of this whole process and go into a bit more detail on that one. Light hits PS1. And again, it gets those electrons excited, and it's a primary pigment, so it releases it to a higher energy level. Now, I haven't got any 4 electrons to replace these lot, but I have got these 4 electrons that are coming down this chain, and there they go. They go in there to replace those 4 electrons. Now, if the plant is low on ATP, these 4 electrons will get passed back, and it will generate more ATP. If the plant has got sufficient ATP, then what it'll do is it'll pass it to another set of electron acceptors, and the very last electron acceptor is called NADP. So the NADP picks up those 4 electrons that have been whizzing down here, if they have not gone back down to the PS2 chain. So you've got a PS2 chain and you've got a PS1 chain. What's at the end of the PS1 chain is NADP, and what's at the end of the PS2? Well, just electron acceptors passing them on to PS1. If you send it back, the PS1 electrons, we call that cyclic, because it's a little cycle. Can you see? Cyclic photo, because it involves light. Phosphorylation, because it means you're making ATP. Okay. Now, what's left behind? Oh, these hydrogen ions. And what they do is they go over here as well. So you've got 4 hydrogen ions, 4 electrons. That means you've got 4 hydrogen atoms. And they join on to NADP. And that makes reduced NADP. So after all that fafadoobie of this light-dependent reaction, what have you made? You've made a bit of ATP and you've made a bit of reduced NADP. And the oxygen is made by photolysis. All right? That is a light-dependent reaction. Practice it so you can draw it out. Now, let's have a look now a little bit more closely about this part, because I think it'll help you with respiration when that comes on. All right, let's have a look at that. So I'm going to draw a bit of a membrane. Okay. And we're passing it down to a series of electron carriers. I'm only going to draw 2 electron carriers. Okay, electron acceptors. Okay, but what I'm also going to draw in is some channels. And I'm also going to draw in a channel. But at the end of the channel, I'm going to draw it attached to an enzyme. And that enzyme is called ATP synthase. Now, what attaches to ATP synthase is ADP and P. They attach to the enzyme. So all they're looking for is the transfer of some chemical energy to join these 2 together to make ATP. So how on earth is this ATP forming? So as the electrons move down, we know that energy is being transferred because it's going down from high energy to low energy. So we know the electrons are transferring energy now. What are they going to do with this energy? Well, what they do is they pump hydrogen ions across the membrane. Okay, and what you create in the stroma, these hydrogen ions are being pumped across this membrane. More and more hydrogen ions are being pumped across. Where are they getting the energy from? They're getting it from the transferred energy from the electron transport chain. This is a series of redox reactions. So the hydrogen ions are being pumped across. They're being pumped across the membrane, the thylakoid membrane, using energy that is being transferred from those 4 electrons as they go down the chain. Imagine you're going down a slide, and you're going down a slide, and that energy that you're transferring is being passed on. And it's causing hydrogen ions to be pumped, and it's creating an electrochemical gradient because you've got high to low. Now, this is where this channel comes in, because now they move down from a high concentration to a low, down an electrochemical gradient. But because they're passing this enzyme ATP synthase, that energy is transferred to joining these 2 together to chuck out a molecule of ATP. It's clever, isn't it? So as the hydrogens come down the electron transport chain, they're transferring the energy to pump hydrogen ions across the membrane to create an electrochemical gradient. The electrochemical gradient comes down through a channel, through hydrogen channels, but it passes ATP synthase, which transfers the energy onto ADP and P to make ATP. Okay. What's great about the light-dependent reactions, you're getting a series of energy changes. So you've got your light is your original source of energy to getting excited electrons. So these electrons, if they're getting excited, they're going to have kinetic energy. Then you're passing that energy in those excited electrons through a series of redox reactions to creating a hydrogen ion electrochemical gradient. And from that electrochemical gradient, with the help of ATP synthase, what you are then generating is ATP. Look at all these energy transfers you're getting. Light energy to excited electrons to a hydrogen ion electrochemical gradient to a molecule of ATP. And that ATP is stable and it can be used when it's needed to in one easy step. So the light-dependent reaction, what has it produced? It has produced ATP and it has produced reduced NADP. Okay, and all that's happened, if it's called reduced NADP, because it's picked up 4 hydrogen ions from photolysis, and it's picked up the 4 electrons from the electron transport chain of PS1. Okay, happy days? And the ATP has come from PS2. Okay, so stop the video there, recap, because that's the end of the light-dependent reaction. We're now going to move into the stroma. So the stroma is this liquidy part, which makes up the majority of the chloroplast. And because it's full of liquid, it's full of enzymes. And there's one enzyme we need to know pretty well, and it's called rubisco. I'll never forget, I wrote Ribena in my A-level for this one, but it's rubisco. And it's annoying because it doesn't end in ASE. But what it does, it likes to join RUBP. Can you see why it's called rubisco? RUB. It likes to join this 5-carbon molecule called RU, I believe Peter presenter now, ribulose bisphosphate, and carbon dioxide together. Okay, do the maths. 5 + 1 = 6. So rubisco catalyzes this reaction, and we call that carbon fixation. Okay. Here we go. Carbon dioxide joins on then with RUBP. 1 carbon plus 5 carbon makes 6 carbon. It's incredibly unstable and it's joined together by rubisco. Love that one. Joins them 2 together, but it makes an unstable 6 carbon. Who do you go and see when you're feeling a bit unstable? You go and see your GP. In fact, you go and see your GP 3 times to get it sorted. Always best to do it 3 times. So the 6 carbon is unstable and it turns into 2 molecules of glycerate 3-phosphate. Glycerate 3-phosphate. Glycerate 3-phosphate. Glycerate 3-phosphate is pretty useless. So what it's done, it's reduced. Please use that term. It's reduced. How do we reduce it? By adding hydrogen. Well, where am I going to get hydrogen from? Well, I get it from reduced NADP. So it gives up its hydrogen and goes back to being NADP. So you're regenerating the NADP, which can go off and can go back into the light-dependent reaction. You need a bit of energy to join this hydrogen on there. You need to break bonds and reform bonds. So we also add the ATP. So can you see what we've got here are the products from the light-dependent reaction. And they're used to turn GP into triose phosphate, a much more useful molecule. Still 3 carbons. Okay. Now, the amazing thing is TP plus TP will make a molecule of glucose. So these two could join together, make glucose, and that's the end. But we need to regenerate the RUBP. I would say that is the single most important step. We have to regenerate the RUBP. If we don't regenerate the RUBP, we can't pick up more carbon dioxide. We can't carry out carbon fixation. So RUBP is really important in that process. So we have to regenerate the RUBP. Please use that in all your answers on this. So how does this actually work? Well, it's got to go around 6 times before we make a molecule of glucose. And the key is this, because TP plus TP contains 3 plus 3, which is 6. But we only need 5 of those carbons to regenerate RUBP. So theoretically, you're chucking out a carbon each time. And if you go around 6 times, you'll have 6 carbons. So you could argue, well, I've got a 6 carbon sugar. Let's just work it out from a molecular point of view as we go around this cycle. So if we've got to go around 6 times, we'll have 6 lots of RUBP and 6 lots of CO2, okay, which will form 6 lots of that unstable 6 carbon. That's a total of 36 carbon atoms, okay. Now that they split automatically into 6 lots of G3P and 6 lots of G3P. Because remember, it's only got 3 carbons. So 6 times 3 carbon molecule is 18 carbons. 18 carbons. Can you see? We've not lost any carbons yet. And then what will those 6 lots of G3P turn into? Well, those 12 lots of G3P will turn into 12 lots of TP. And again, we're not losing any carbon. Now, this is the magic bit, and you know that that's reduced NADP, turning back into NADP. And you know that ATP is giving up its energy and turning into ATP plus P. TP plus TP equals glucose, which is 6 carbons. Now, if I use 2 of these TPs to make a molecule of glucose, how many TPs will I have left? Well, I've got a total of 12 lots of TP. Would you agree with that? I've got 12 lots of TP, 12 molecules of TP. If I'm using 2 lots of TP to make the glucose, that leaves me 10 lots of TP, which is 30 carbons, because each TP is 3 carbons. 10 times 3 is 30 carbons. That means I've got enough to make 6 molecules of RUBP. Because RUBP is 5 carbons, 6 times 5 is 30. Yay, balanced. So that's what happens. It goes round 6 times. It goes round 6 times. And what you're left with, if it goes round 6 times, you will have 12 molecules of TP. And those 12 molecules of TP, 2 will be used to make 1 molecule of glucose. And the rest, 10 molecules of TP, will be used to make 6 molecules of RUBP. And the term I'd love you to use is the regeneration of the RUBP. We call this Calvin, the Calvin cycle. And it is a legend of a carbon cycle because it has to go around 6 times, getting a bit dizzy, making 1 molecule of glucose. So let's get it. 6 lots of RUBP, join on 6 lots of CO2 to make 12 lots of G3P, which makes 12 lots of TP. 2 lots of TP make glucose. And what happens to the other 10? The 10 lots of TP go on to make the 6 lots of RUBP. How many atoms of carbon in a TP? 3. If I've got 10 times 3, I've got 30 carbons available in that 10 lots of TP. RUBP is 5 carbon molecule. 6 times 5, 30 carbon. Life is sweet because we balanced all our carbons. What's fascinating about this is you need the products of the light-dependent reaction to turn GP into TP. And that is a major focus of exam questions. What happens when you don't get the light-dependent reaction? Because you've turned the lights off, it's called nighttime. So what happens when your amount of ATP, your amount of reduced NADP goes down? Well, then your GP can't get turned into TP, but TP still gets turned into RUBP, which picks up carbon dioxide. It's just there's a backlog. It's like the GP, the G3P builds up. It can't cross over to TP because you've not got any of the products of the light-dependent reaction. Folks, I would practice drawing these reactions, if you can. You can draw these really quickly. You could do something like that, whoosh, going down, in, back over, going down, NADP. Okay, so you could have something like that. And you've got that being chucked out. And you've got this. That goes there. That replaces, that replaces those. PS2, PS1. Okay, that replaces that one. And the oxygen, well, that goes into you, me, the hamster, the tortoise, whoever wants it. Okay, so what do we have? We've got reduced NADP, and we've got ATP, and they go off into the light independent reactions. So we've got RUBP, which is 5, joining on CO2, which is 1. We call that carbon fixation. It's carried out and catalyzed by that beautiful enzyme called Rubisco, and that turns into 6 carbon, unstable. Do you know what? Sometimes I don't even talk about that 6 carbon. And it turns into 2 lots of G3P, which turns into TP using the products, so the ATP and the reduced NADP. And then the TP plus TP, it goes back to making the RUBP. But if it goes around 6 times, you get 12 lots of TP made, folks. And you know that 2 lots of TP will make 1 molecule of glucose, and 10 lots of TP will make 6 lots of RUBP, which means that they can pick up 6 lots of carbon dioxide. So 6 is the magic number. I hope that has helped. Please practice drawing it out. Make your diagram simpler each time. And I hope you've enjoyed photosynthesis, because do you know what? It's enjoyed providing you with the glucose. And do you know what? It doesn't just leave it at glucose. We always talk that glucose is made by a plant. But do you know what? That glucose can be polymerized into starch and cellulose, which is important for storage and making cell walls. But do you know what? That glucose can also have nitrogen added to it and turn it into an amino acid for your proteins. It can have more hydrogen added to it, so it can be turned into fatty acids. Do you know what? With that glucose, it can make a wide range of organic molecules, and we call this product synthesis. Plants are clever. They make organic molecules. Have a good one. Practice drawing it out. Email me over it. Love to see them. Take care now.